new study conducted by researchers at the University of Oxford has challenged previously held views that brain preservation in the archaeological record is extremely rare. The team carried out the largest study to date of the global archaeological literature about preserved human brains to compile an archive that exceeds 20-fold the number of brains previously compiled. The findings, entitled Human Brains Preserve in Diverse Environments for at least 12,000 Years, were published in the journal Proceedings of the Royal Society B. Soft tissue preservation in the geological record is relatively rare and, except where deliberate intervention halts the process of decay, for instance during embalming or freezing, the survival of entire organs is particularly unusual. The spontaneous preservation of the brain in the absence of any other soft tissue, that is, the brain's survival amongst otherwise skeletonized remains, has historically been regarded as a one-of-a-kind phenomenon. The new research reveals, however, that nervous tissues actually persist in much greater abundances than traditionally thought, assisted by conditions that prevent decay. The study, led by postgraduate researcher Alexandra Morton-Hayward of Oxford's Department of Earth Sciences, brings together the records of more than 4,000 preserved human brains from over 200 sources across 6 continents and in more than 10 languages. Many of the brains were up to 12,000 years old and found in records dating back to the mid-17th century. These shrunken, discoloured tissues were found preserved in all manner of individuals, from Egyptian and Korean royalty, through British and Danish monks, to Arctic explorers and casualties of war. Scouring the literature and canvassing historians worldwide, the detailed search found a dizzying array of archaeological sites yielding ancient human brains, including the shores of a lake bed in Stone Age Sweden, the depths of an Iranian salt mine around 500 BC, and the summit of Andean volcanoes at the height of the Incan Empire. Every brain in the database was matched with historic climate data from the same area to explore trends in when and where they were found. The analyses revealed patterns in the environmental conditions associated with different modes of preservation through time, including dehydration, freezing, saponification or the transformation of fats to grave wax, and tanning, usually with peat, to form bog bodies. Co-author Professor Erin Sorp of the university's Department of Earth Sciences said, "This record of ancient brains highlights the array of environments in which they can be preserved, from the high Arctic to the arid deserts." Over 1,300 of the human brains were the only soft tissues preserved, prompting questions as to why the brain may persist when other organs perish. Interestingly, these brains also represent the oldest in the archive, with several dating to the last ice age. The mechanism of preservation for these oldest brains remains unknown. However, the research team suggests that molecular cross-linking and metal complexation, proteins and lipids fusing in the presence of elements such as iron or copper, are feasible mechanisms by which nervous tissues might be preserved over long timescales. Study co-author Dr. Ross Anderson said, "These ancient brains provide a significant opportunity for unique insights into the early evolution of our species, such as the roles of ancient diseases." Alexandra Morton-Hayward, lead author of the study, said, "In the forensic field it is well known that the brain is one of the first organs to decompose after death, yet this huge archive clearly demonstrates that there are certain circumstances in which it survives. Whether those circumstances are environmental or related to the brain's unique biochemistry is the focus of our ongoing and future work. We're finding amazing numbers and types of ancient biomolecules preserved in these archaeological brains, and it's exciting to explore all that they can tell us about life and death in our ancestors." The university says that finding soft tissues preserved is a bioarchaeologist's treasure trove. They generally provide a greater depth and range of information than hard tissues alone, yet less than 1% of preserved brains have been investigated for ancient biomolecules. The mind literally boggles at what they might reveal. The untapped archive of 4,400 human brains described in the study may provide new and unique insights into our history, helping us to better understand ancient health and disease and the evolution of human cognition and behaviour. But that's it for this video. Don't forget to like, share, and most importantly subscribe. And you can also support the channel on Subscribestar via the link in the description, or via YouTube Super Thanks. Thanks for watching. Bye for now.